Good morning, everyone. Thank you for joining us this early for the second breakfast seminar of Biognosys this week. I'm Fabia. I work in the business development of the Biognosys products, and I am delighted to chair today's session. We have two great speakers who will tell us about how Spectronaut 16 is the best ally for the visionary projects. I will first give you a short introduction about Biognosys and our products portfolio. I will then hand over to Jesper Olsen, professor at University of Copenhagen. He will talk about a novel hybrid DIA acquisition strategy that combines targeted and discovery proteomics. Our second speaker is Yansheng, professor at Yale University School of Medicine, and he will present about rewiring of the proteome and phosphoproteome in mammalian cells under selection and stress. So Biognosys was founded back in 2008 as a spin-off company from the group of Professor Rudi Ebersold at ETH in Zurich, and has been leading proteomics innovation for over 13 years. Thanks to a strong scientific foundation and R&D-centric mindset. At present, we have many international patents, and to date, over 2,000 publications feature Biognosys technology. We also are one of the largest facilities in the world for large-scale discovery proteomics. This allows us to serve our ever-growing global customer base thanks to our contract research services as well as products. On the product side, we do have, on the one hand, our software solutions, and Spectronaut is our flagship software for DIA. We also have SpectroDive, our software for targeted proteomics, and SpectroMine for DDA proteomics. Last but not least, we also have our free software Quick for quality control. And Quick works together with the IRT kit, and when you spec the IRT kit in your QC sample, you get near real-time QC readouts. And so you can track instrument performance. We have PQ500, our kit for the absolute quantification of over 500 proteins, plasma proteins, and the sample preparation kit for seamless and high throughput sample prep. So Spectronaut allows the analysis of DIA data in different ways. We do have a more traditional library-based approach, but also our library-free workflow, direct DIA. SpectroDive supports different targeted proteomics data type, MRM, PRM, and also thermal SureQuant. And SpectroMine, our software for DDA analysis, is particularly ideal for isobaric labeling quantification, ILQ. When developing our software solutions, there are three central concepts or principles that are very important for us that we always want to aim to. The first thing is performance that translates into speed, but also identifications. Reliability. For us, it's very important to have a robust scientific solution that is trusted by the community. And ease of use. I think that all of you who are already users of our software solutions can appreciate that they are incredibly intuitive and friendly and easy to use. And so if you haven't tried our software yet, you can. We do offer trial licenses, during which we do provide full support on data analysis, on the best analysis settings, and more. And so if you are interested, please visit us at booth 306. So I would like to spend just a few words on Spectronaut 16 that we released last Monday. We had a launch seminar. If you missed it, no worries. We have it recorded, so it's going to be available on demand. And the development of Spectronaut 16 has been heavily centered around the implementation of artificial intelligence that allows to extract much more from the data. So I think I can say that probably at the moment Spectronaut is the most AI-augmented proteomics software out there. And this allows with any workflow, both library-based as well as library-free, to really reach new depths from the data. Since both our speakers are going to present phosphoproteomics work, I also would like to stress here that Spectronaut is particularly good when it comes to PTMs projects. And so on PTMs, I stop and I leave the stage to Jesper.
Okay, so thank you. Can you all hear me? It works out fine? Okay. Yeah, so first of all, thanks for the invitation to come here and give you a little insight into some of the work that we've been doing in my lab over the last couple of years. Today I'll present a novel hybrid DIA acquisition strategy where we combine targeted and discovery proteomics, and this is a close collaboration with Thermo Fisher Scientific, and in particular Carl Ford, who's actually here in the audience today, did all the programming. And then from my lab side, it's Anna Martinez Del Val, who worked to test and apply this strategy. But in my group, we have, of course, a huge interest in applying quantitative phosphoproteomics, and we've really been trying to develop short gradients and fast acquisition strategies and with DIA. And we are, of course, really excited, especially with the direct DIA workflow, where we can get really good coverage in a relatively short period of time without having to go through the pain of acquiring these project-specific libraries that otherwise would be needed to do the DIA workflow. The problem, of course, with DIA, as with any other method, is that you are not always guaranteed that you will identify the same peptides when you go through hundreds of samples. And although we have really tried to optimize the conventional DIA workflows to maximize coverage and minimize the number of missing values, then of course for really low abundant phosphopeptides, for example, it is indeed better often to make like a use of a targeted method such as SureQuant, where you can be confident that you will get very accurate quantitation for your peptides of interest over hundreds of conditions. But of course, if you do a targeted method, you may miss out on all the other phosphorylation sites that you may have in your sample that you're not targeting. So that's why we thought it could be very interesting to try to see if one could make like a hybrid workflow where you would be able to do both a targeted analysis while also at the same time perform the unbiased discovery analysis with the standard DIA method. And basically what enables this is then API in the Tune software, where you can do intelligent data acquisition using isotope labeled internal standards that you spike into the sample that can then, when detected on the fly, can trigger multiplexed PRM spectra, where you can then select a very narrow isolation width and of course maximize the ion injection time of the endogenous peptide, while you simultaneously also then acquire DIA data of the full proteome or phosphoproteome of interest. And the basic principle is that you spike in heavy label standards. If such a peptide is detected in your full scan, it will then trigger a fast PRM scan of the heavy peptide. If a predefined set of fragment ions are matched and detected, it will then go on to do a multiplex PRM scan where it will record for both the heavy and the light peptide in the same run. And the main advantage of this is that then you can use differential injection times such that you can basically normalize the ion abundance of both the heavy and the light peptide. of course, you have to then correct for that afterwards, because typically you are in the situation where the heavy synthetic peptide is present in much higher abundance than the light version. So you would have to fill up for much longer time with the light than with the heavy. And afterwards, of course, that you need to take that into account when you do the quantification and of the peptides. And right now, Anna had to make a quite elaborate workflow to analyze this type of data. So, of course, for the regular DIA part of the file, we just use Spectronaut for the analysis. But for the multiplex scans, we actually have to go through quite an elaborate workflow in order to extract the multiplex scans, convert them into an MCML file, import them into Skyline in order to export transition list, raw retention intensities and retention times, and then do afterwards the injection time correction and quantification. And we are, of course, very eager and interested to see if maybe all of this could be implemented in SpectroDive in the future, but let's see what will happen. But in the meantime, Anna actually wrote a very nice piece of software where we can do all these corrections, and we can actually also quantify over the different conditions, the individual peptides of interest that we have spiked into the sample, such that we can directly from the data that we get out of Skyline. So in order to stress test and see how applicable is actually this hybrid DIA workflow, we've then made use of this multi-pathway phosphopeptide standard kit that is traditionally used in SureQuant, where you're basically monitoring 7 different signaling pathways at the same time. So you can see a list of them here. And it's a little bit more than 130 different phosphopeptide standards that we can then spike into our sample and try to monitor. And we can see that we can typically detect more than hundreds of these peptides with the workflows where we make use of the Jesper methods on the EvoSep1. And we are doing all of the measurements in the Explores 480 mass spectrometer. And we can see that in general we get very, very good quantification both from the heavy peptides and from the endogenous peptides when we use these relatively short and very sensitive gradients. And of course, one of the things that's important is to really benchmark this method directly against both conventional DIA and also against SureQuant. And in this case here, we can see that it's basically 129 different heavy peptides that we can detect with the 20 samples per day method. And actually, with the hybrid DIA, we seem to be able to identify the endogenous peptides, roughly 100 of them throughout the analysis when we just spike them into really low amount of HeLa peptides. So we get really high sensitivity all the way down to 6.2 microgram of input material before we do the phosphopeptide enrichment. Yeah, and basically we can see when we compare to SureQuant and to conventional DIA that for high abundant peptides, of course, all methods perform really well and you can get good quantitative readouts for all of the peptides. But as soon as you go to more peptides in the medium abundance range and in the low abundance range, those are either not detected at all or quantified very poorly with DIA, but both with hybrid DIA and with SureQuant, we usually get good quantification across the different input amounts. And we can see here that it's, for example, only at the high abundance peptides that with conventional DIA that we're able to detect the endogenous peptides at all different concentrations, whereas at medium abundance and low abundance, it's only SureQuant and hybrid DIA that are able to do the quantification. So we thought that we wanted to then apply this to a typical signaling study, and in our lab we have focused on EGF receptor signaling for many years. So we thought that it could be interesting to see if we stimulated HeLa cells with EGF for different time points and used inhibitors of some of the downstream signaling pathways. Can we then monitor the quantitative differences between the different signaling pathways as they are turned either on or off with the inhibitors? And then to do this, we started with 3 to 4 million HeLa cells. We either stimulated them for 10 minutes or 90 minutes with EGF in the presence or absence of either an EGFR inhibitor, a MEK inhibitor, or a PI3 kinase inhibitor. And then we went through our normal workflow, do the PAC digestion, spike in the phosphopeptides, do the phosphopeptide enrichment, and then analyze everything on the EvoSep and Exploris 480 using both SureQuant and hybrid DIA. And we're really happy to see that for all the phosphorylation sites that are regulated by EGF or MEK or PI3 kinase specifically, we see essentially identical data with both hybrid DIA and SureQuant. And it's probably easier to see here when we look at the raw ratio between light and heavy, where you have in the purple ones you have SureQuant, and in blue you have hybrid DIA. And you can see in general they are basically right on top of each other throughout all the different peptides that we are monitoring that are regulated either by EGF itself and then inhibited when in the presence of the EGFR inhibitor, or inhibited by PI3 kinase but still activated by the MEK inhibitor. And the advantage, of course, of this type of strategy is that you can then also just analyze all the background data that you have in the file in order to get the full phosphoproteome. In this case here, we could then identify a little bit more than 5,000 phosphorylation sites with a high localization site probability. And when we perform quantification and kinase substrate enrichment analysis, we can see, as expected, that, for example, the MAP kinase pathway are activated by EGF. It's completely absent in the presence of the EGFR inhibitor and, of course, in the presence of the MEK inhibitor, whereas it's also fully activated by in the presence of the PI3 kinase inhibitor. And conversely, with mTOR, we can see that it's down with EGFR and it's down with the PI3 kinase inhibitor. So with this, I hope I've been able to show you that we have a very good hybrid DIA workflow now that really combines targeted and discovery proteomics. We do this heavy spike-in standards that can then trigger these multiplexed PRM analysis with simultaneous DIA analysis of the rest of the peptides. We have the computational workflow now for processing, quantifying, and visualizing these hybrid DIA scans. We get data that is comparable to the SureQuant strategy in terms of quantitative accuracy for the targeted peptides. And we can really monitor the specific EGFR pathway status, especially with this multi-pathway phosphorylation kit, where we can cover the activation status of 7 different signaling pathways. We can do very sensitive phosphoproteomics down to low microgram of input material. And we are, of course, currently testing this in some different cancer signaling cell models where we're looking at single spheroids. And with this, I would just like to acknowledge the people that did the work. So this was really a project in my lab that was spearheaded by Anna Martinez Del Val. So this is Anna here. And then in close collaboration with Thermo Fisher Scientific, Alexander Makarov's team, Yui Suan, and Kyle Ford. And of course, we're very happy with the close collaboration that we have with Lukas Reiter's team on the Spectronaut for phosphoproteomics. And with that, I would like to thank you for your attention, and I'll be happy to take questions if time allows. Thank you.
We have a Q&A session in the end, so save your questions.
Okay, fine.
And we'll do that together in the end. Good. Thank you very much, Jesper. Our next speaker, Yansheng Liu.
Hello, everyone. Good morning. So I feel honored to speak after Jesper. And today I'm going to show you more about the application side. So my title today is Rewiring of the Proteome and the Phosphoproteome in Mammalian Cells under Selection and Stress. For some of you may know that my lab at Yale is already 4.5 years old, and there are essentially 3 research areas that interest us. The first one is the DIA mass-based methodology development and also bioinformatic tools. And the second is the impact of the post-translational modifications, such as phosphorylation on protein turnover and cancer signaling. And the final one is the biological variability in different scales. So this research interest actually stems from my previous postdoc project and research result with Rudy Episode group when I was doing my postdoc. So regarding this biological variability, what I'm quite excited about is that, as you see, we wrote this review a few years ago together with Andreas Bayer and Rudy, and we essentially discussed the different scenarios of mRNA protein correlation. And we also point out that the variability at the proteome level can happen at different scales. The first is the one genome, one individual scale. The second is inter-individual, and the third is interspecies. So for the one individual study, I quickly show you a paper from Mike Snyder group where they look at the different tissue proteome and quantified the biological differences between these tissues on the protein level. So one genome can code quite a diverse proteome. If we define the one genome as one individual, I would reasonably argue that the paper we published a few years ago in these monozygotic twins also can be counted as a one individual study. So in this particular project, we isolated the fibroblast cells from the monozygotic twins that are discordant for trisomy 21. So basically, one of these cells bears another chromosome 21 copy. And then we can quantify by that time by source and also pulse-like DIA to look at the proteome and the protein turnover level. And we discovered many interesting rules. For example, a principle that can govern the protein level dosage compensation. So here you can see that the mRNA or the transcript that are encoded from the chromosome 21 are all regulated by 1.5-fold change. However, at the protein level, the reaction is quite heterogeneous. You can find if the protein participating protein complex, we can see the fold change is buffered back to 1-to-1. And we somehow ascribe this to the accelerated protein turnover rate. So basically, this little bit busy box plot tells you that there is essentially a protein level buffering effect mediated by protein complex stoichiometry that will help us to understand the prototype in aneuploidy cells, which can be important for cancer. So in the same study, we also performed the inter-individual analysis. So here we isolated 11 fibroblast cells, and then we can confirm this complex-based buffering effect. We can also try to correlate the one genome study together with this multi-genome or multi-individual study, and we find that, for example, the downregulation of the mitochondrial proteome is actually a hallmark in human Down syndrome. So this is all good, but till now, there is one component that is essentially missing that we didn't contribute by our favorite DIA tool, that is the interspecies diversity. So therefore, before talking about that, I will also show you quickly something about the human plasma project, because in this plasma project, we actually look at the plasma proteome that are important for biomarker discovery, as you guys know, and we indeed enlarged the sample numbers in this cohort to 232 human twins. So here we also quantified 300 plasma proteins, and then we quantified the origins of variability into different genetic component, environments, and aging effects. So this is another inter-individual study. So one thing that I will share with you is actually an interspecies study. So that is something that we are quite excited. In the last 2 years, we collaborated with evolutionary biologist Gunther Wagner, who also had a lab at Yale, and we finished this data set where we isolated the skin fibroblast cells from different mammalian species, and then we quantified the proteome by DIA mass and also used the FE-IMAC workflow to enrich the phosphopeptide and quantified the phosphoproteome. So we used, of course, Spectronaut to control the peptide level FDR, protein level FDR, and also used the PTM localization score to report the P-site level result. And these 11 mammalian species essentially are divided into two groups, the ant group, which contains the rabbit, rat, monkey, and human, and also the lout group, which contains the sheep, cow, pig, dog, and cat. We also have Olsen as an out group. So from this data set, we get a quite nice coverage at the proteome and the phosphoproteome level. And at the phosphoproteome level, we can quantify 8,000 phosphosites. So this may look a little bit lower than what we reported in Barbara Savaska's talk, my postdoc's talk in this Monday, where we report 50,000 phosphosites in cancer cell lines. This is because we actually started with a much lower sample amount. But still, we have a pioneer data set where we can map this gene orthologs and quantify 1,600 proteins across the species, as well as 546 phosphoproteins corresponding to more than 600 phosphosites in all of these species. And in the heat map, you can see we achieved very good biological variability per species. So from here, we can address some questions. For example, we can use PCA to look at the diversity or variability at each level and use paired mRNA and protein data. You can see that transcript can actually split the two groups of the mammalian species, while at the protein level, there are more variability. And at the phosphosite level is even less unclear, but this could be related to the limited coverage of the phosphoproteome we achieved. For the opsome, which is the out group, we can see that indeed it is distant from the other two groups. Therefore, there are some biological variability captured in our data for sure. And then we want to look at the mRNA protein correlation at the absolute scale because of our long-term research interest in mRNA protein correlation. So here, the mRNA protein Spearman correlation is determined to be 0.6 across all of these 11 species. This essentially suggests that the mRNA primarily determines the protein level variation and abundance to the similar extent in all of these mammalian cells, which is consistent to the previous human-based or mouse-based work. So we also look at the gene-specific correlation, which is more interesting for sure than the absolute level correlation. And here you can see across the species, we can quantify the correlation between mRNA and protein and between phosphosite and protein. And we find certain geopathways has a higher correlation across species than other geopathways. And also for the protein phosphorylation correlation, we can see, for example, the cellular component movement have a much lower across species correlation. So this essentially suggests that there is an extensive post-transcriptional level regulation as well as the phosphorylation-mediated regulation among these species. So we then want to quantify this. We get an estimation, which is the SD, the standard deviation of the log2 transformed values at the mRNA and protein level, and correlate that to the gene functions. And we find, for example, the essential genes has a much lower variability than the non-essential or conditionally essential genes. And we also find the younger genes have a higher variability than these older genes shared across the eukaryotic cells. And we also look at the cancer secretome, the surface proteins, and the biomarkers, and we find that, for example, here, the secretome proteins are having a much higher variability at the both mRNA and protein levels. So essentially, our data provide an evolutionary angle for understanding gene functional diversity. So here we want to look at this in an unbiased way. Therefore, we used this 2D notation enrichment analysis where we could correlate the variability at the protein level and the transcript level. And we find that, for example, these extracellular matrix processes had a much higher variability at both levels, while the proteosome complex or the phosphosite complex, these correcting machineries, seems to have the least variability. They are very stable across these species. Also, we find that the translational related processes are particularly stable at the protein level, suggesting that they can be very important for protein level buffering effect during the evolution. So then we asked the interesting question, how can we understand the interspecies diversity with the reference to the inter-individual diversity? So therefore, we used this data that I was telling you about because we were also analyzing fibroblast cells, and we have the DIA mass spec measured for both data sets. So it's very safe for us to incorporate the 11 individual data set and the 11 species data set together using the PCA analysis. And here you can see, as expected, the human individuals are much more close to each other compared to the other species levels. However, these protein complex level results are more interesting because you can see that at the across species level or across individual level, we can see that the complex in proteins showed a lower variability than the complex out proteins. However, at the transcript level, previously at the individual scale, we actually didn't see a significance between these two groups. However, now there is a significance. So we think that this could suggest that the evolutionary process already started to shape the transcriptome expression towards the need at the protein level. So we are quite interested to see this result. We also did this 2D enrichment analysis now to understand the variability at the inter-individual scale and the interspecies scale. So we can see that, again, interestingly, the extracellular matrices are showing a higher variability, while the proteasome least variability. And more interestingly, we can see these RNA metabolism processes and cell division are showing a particularly higher variability across species, suggesting that they may be very important during mammalian evolution. So if we look to these two of these 2D enrichment analyses together, we can have some quite fundamental understanding about biodiversity, because these interspecies and inter-individual diversity are the foundation for the biodiversity on Earth. So here you can see on the left, we have the post-transcriptional diversity. On the right, we have the prototype diversity across individual and across species. We can find some universally variable processes and also universally stable processes. So we also look at the phosphosite. We didn't have a great number because if we do the sequence matching, we only have 600-ish phosphosite. And we did a regression-based normalization, and we can see that even after that, the phosphosite showed a higher variability than the protein level and the transcript level, suggesting there are more information in the phosphoproteome, which is good news for us. And we also look at the score distribution, like this Bichao score that published a functional score, and you can see the functional score for this commonly identified phosphosite across species, of course, higher than others, and also get more conservative than others, which is kind of expected. From there, we look at this phosphosite co-variability. We try to elaborate a co-evolutionary phosphosite. Here in this network, you can see each node represents a phosphosite, and the link represents a positive or negative co-variation. And you can see this network is very intensive, and also 95% of these links are actually in the positive range, suggesting that there is a strong co-evolutionary pattern for phosphorylation in these mammalian cells. We look at the motifs as well, and we find these motifs, many of them can be annotated as CDK substrates. Together, this actually also supports some biological annotations in the network. So finally, we look at this protein degradation machinery diversity, because as I was showing you, proteasome showed the very high stability, but there are a few proteasome subunits, like this immunoproteasome component are actually showing a higher variability than other components. What is even more exciting to us to see is that we also look at the lysosome, the deubiquitin enzyme, and the E3 ligase, and we see this variability are actually much higher than the proteasome level. So as we know, there are two major machineries in the mammalian cells, like the proteasome-based and the lysosome-based protein removal degradation process. And it's kind of interesting for us to confirm that the proteasome is quite conserved during evolution, while the lysosome showed a higher variability. And we independently searched the data towards the ubiquitination modification using the signature peptide for the ubiquitin sequences. And you can see, interestingly, the K48 is showing ubiquitin linkage is showing a higher expression abundance and also lower variability. So because the K48 is known to mediate the proteosome-mediated degradation, so these two levels of data agrees very well, telling us that there is a variability component in protein degradation machineries. So I will skip this conclusion, but I just want to quickly mention that the biological variability across these individual and the species can be very important for us to think about and to measure more in the future. So in the final 3 slides, I will switch gears quickly and talk about the plasma proteome, as I was trying to include an additional slide during introduction to say the plasma proteome is very important. And here we have like 4 samples that we tested. We did an unfractionated human total plasma DIA data set. And essentially we want to ask if we compare Spectronaut 16 and 15, whether or not we could identify more proteins. We used a small library, which we downloaded from this PNS paper using several files from them, and they already identified more than 1,000 plasma proteins. So use the same library, the same data set, we can see that the Spectronaut 16 gives us 20% more identification at the human plasma level without fractionation. So we then want to push this. We want to see if we have a much larger complete library, like this full library. Can we still identify more? And here we actually downloaded all of the files from this PNS paper together with some of our local DIA-mass data sets. And we have a library that we call a full library containing 1,800 proteins. By using this library, and we compare to the small library result in the previous slide, as well as the direct DIA, which is library-free. And you can see we get almost 20% increase compared to the small library and almost a 70% increase compared to the direct DIA result. So this suggests that Spectronaut 16 in our hand can be very useful in the library-based workflow where you have a large library and then you get much more from your same data set. So finally, I want to make the remark that nowadays with the Spectronaut and with all this instrumentation, compared to what I reported 7 years ago, we can get twice as much at the peptide and protein level for this very challenging human sample of plasma proteome, but very important for clinical biomarker discovery. So with this, I want to thank my small group at Yale, especially the previous postdoc Chen, who did the evolutionary biology work together with Gunter's group, and Wenxue, who is here, who generated the plasma data, and Barbara Savaska, who is here and also in the audience, who talked to you about the colorectal cancer cell line data set in her Monday talk under the treatment of metformin, for which I didn't have time to introduce today. So that is related to the stress part in my title. So also my funding support, and also Akiko's group for sharing the plasma data set with us. And of course, my friends and colleagues in Spectronaut team, in Biognosys, Tejas and Lucas. I'm happy to see your success in the last 13 years. I was witnessed the first version of Spectronaut, and now it's version 16 after 13 years. So it's very exciting, and congratulations. And thank you for your attention.
Thank you very much. Let's give a last final round of applause to our speakers. And enjoy the rest of the day.